Okay, now we'll understand the action potential that is generated at a muscle and overall structure of a muscle. So when we talk about muscle, it consists of bundle of myofibrils. So there are numeral myofibril, and the bundle of myofibril, they actually constitute muscle. Now, if we look at myofibril in detail, in close detail, so basically myofibril, they are surrounded, they are actually covered by T-tubules. So in blue you can see T-tubules. These are T-tubules, and they're covered by sarcoplasmic reticulum as well. So there are T-tubules and there's sarcoplasmic reticulum. Now these T-tubule and sarcoplasmic reticulum, they're arranged in a triad fashion. Now what is this triad fashion? There's a sarcoplasmic reticulum, then a T-tubule, and then another sarcoplasmic reticulum. So basically a T-tubule, it is present alongside two sarcoplasmic reticulums. Point where or the relation of sarcoplasmic reticulum or attachment of sarcoplasmic reticulum to T-tubule is through cisternae. Okay. Now this was outer structure of myofibril. But when we go inside this cell, thick and thin filament are present inside myofibril. So myofibril is covered by T-tubules and sarcoplasmic reticulum, but inside myofibril, thick and thin filament are present. So now we study these thick and thin filaments. Okay, so this is thin filament, this is thick filament, right? Thick filament is basically made up of myosin. This is myosin, and it has two heads, right? So it has two heads, and then it is composed of myosin. With these heads, ATP can bind, and this head, it also has a role in binding to the actin of thin filament during contraction or excitation contraction coupling. So during excitation contraction coupling, basically this binds to actin of thin filament. Okay, now we talk about thin filament. So thin filament, basically it is composed of actin, but beside actin it also has two other components, troponin and tropomyosin. So what is function of tropomyosin? Tropomyosin makes sure that actin does not bind to myosin. So normally when muscle it is at rest, it is important that actin does not bind to myosin. It is during excitation contraction coupling that actin and myosin binds, and during resting phase of muscle, actin and myosin should not bind, and that is the function of tropomyosin. When we talk about troponin, so troponin is of basically, if we talk about troponin, so we have three types of troponin. There is troponin C that basically binds to calcium, and as it binds to calcium, it removes tropomyosin. Where was tropomyosin present? Basically, tropomyosin was present between actin and myosin, and was making sure that they do not bind. So when troponin C it gets activated because of calcium, it makes sure that tropomyosin it is removed from its position, and actin and myosin they can bind or interact with one another. Then we have troponin I that actually causes inhibition of the coupling of actin and myosin. So it inhibits them. And last we have troponin T. Troponin T basically it binds troponin to tropomyosin. It links troponin to tropomyosin. Okay, so if we see myofibril under microscope, we'll see alternate dark and light bands. These dark and light bands are known as A bands and I bands. So these A band and I band with respect to thick and thin filament actually correspond to certain positions, so you'll understand that. So under microscope we see that certain areas they are dark, and those dark areas are A band. And if you look at A bands in more detail, we see that basically A band majorly they consist of thick filament, and then some part of thin filament that forms A band, right? And I band basically it consists of majority part of it is basically made up of thin filament. So there's a line that passes through center of A band. So the line that passes through center of A band is known as M line, and the line that passes through center of I band is known as Z line. And it is between two Z lines. So it is basically between two Z lines that a sarcomere is established. What is a sarcomere? It is the area of myofibril between two Z lines, and this is the basic contractile unit of a muscle. Okay, now we will discuss T-tubules. We had already discussed what are T-tubules. T-tubules were those structures which were present in triad-shaped fashion, and they were around the myofibril. Now these T-tubules, they are connected to sarcolemma. What is sarcolemma? It is the membrane of this muscle, myofibril. The outer membrane of myofibril or muscle is sarcolemma. So when sarcolemma depolarize, this depolarization will be carried by T-tubules. T-tubule will carry this depolarization to the interior of cell, to the inner, actually what this depolarization will do, that it will cause the conformational chain in dihydropyridine receptors. Okay, what happens that inside T tubule dihydropyridine receptors are present. Okay, now these dihydropyridine receptors which are present inside T tubules, as T tubules will depolarize, because of depolarization of T tubules, dihydropyridine receptors, they'll undergo conformational change. As they will go conformational change, they will affect the receptors of endoplasmic reticulum, because this is T tubule and a T tubule is in very close association with endoplasmic reticulum. So T tubule receptors, when they will undergo conformational change, they will affect receptors of endoplasmic reticulum. The receptors of endoplasmic reticulum are known as ranitidine receptor. Now ranitidine receptors, basically, when they will be affected, they will release calcium. Calcium will be released. So basically, sarcoplasmic reticulum, it stores as well as releases calcium. How it releases this calcium? That is through ranitidine receptors. Okay. So now, when all of the concepts of these different structures of muscles of myofibril have been cleared, now we will understand excitation, contraction, coupling. So what is excitation, contraction, coupling in skeletal muscles? So action potential, it arrives at sarcolemma. From sarcolemma, this action potential or this depolarization is carried by T tubules. Depolarization of T tubules will cause conformational change in dihydropyridine receptors. These dihydropyridine receptors will affect ranitidine receptors of sarcoplasmic reticulum. As they will affect the ranitidine receptors of sarcoplasmic reticulum, they will release calcium. As this calcium will be released, this calcium will bind with troponin C. We have already studied that what is troponin C. Troponin C, if it activates, it removes tropomyosin from between actin and myosin, right? So as troponin C will be activated, this will cause coupling of actin and myosin because tropomyosin is removed. Okay, so let's see here. Now look at this diagram number 1. Troponin C is activated. As troponin C is activated, tropomyosin has been removed. That was causing inhibition of the coupling of actin and myosin. But now this myosin and this actin, they can couple. This is known as rigor state, because this actin and myosin, they will stay coupled. They have a very firm bond. They will maintain this bond unless this myosin be acted upon by ATP and bond broken. So what will happen? ATP will come. This ATP will bind with myosin head. When ATP will bind with this myosin head, it will cause conformational change in this myosin head, and because of which this coupling will break. As this coupling will break, here ATP will hydrolyze into ADP and inorganic phosphate, right? ATP has hydrolyzed, and now what will happen? If let's say if this is position 1, right? So if this is position 1, initially this myosin head, it binds with actin at position 1. Now in second step, when ATP is removed, now it will move forward from position 1 to position 2. It will now bind to position 2. Then it will again bind to position 2, and again rigor state will be achieved, and again ATP will have to come to break that rigor stage. Again, when ATP will hydrolyze, it will move from position 2 to position 3. This is known as cross-bridge cycle. This cycle will carry on. This cycle will continue, and because of this cross-bridge cycle, basically there will be forward movement of myosin. Myosin will jump from position 1 to position 2 to position 3. So myosin will keep on moving, or it will keep on walking along the actin filament. Because myosin will keep on walking along the actin filament, this contraction or excitation, contraction, coupling will continue. How long will it continue? It will continue as long as calcium is bind to troponin C. Now this is very important. Unless this calcium is removed from this troponin C, this whole process of cross-bridge cycle will continue. So how to end this process? There is an enzyme, SERCA, right? SERCA will cause reuptake of calcium by sarcoplasmic reticulum. When sarcoplasmic reticulum will reuptake this calcium, as a result, intracellular calcium will decrease. When intracellular calcium will decrease, ultimately troponin C will break its bond with calcium. This bond will be broken, and when troponin C and calcium, their coupling will be destroyed. Ultimately, tropomyosin will once again come between actin and of course myosin head. Tropomyosin will come back, and when tropomyosin will come back, once again their coupling will be inhibited. So this is how muscle contraction happens. Now what happens in tetanus? In tetanus, there is repeated stimulation of muscle fiber. Because of repeated stimulation, too much calcium is released. When there is a lot of calcium, it means troponin C will remain active for longer period of time. If troponin C will remain active for longer period of time, what does that mean? It means that overall contraction of muscle will last longer and there will be no relaxation. So this is all about tetanus.